I've mentioned a couple of times how these myofilaments are arranged in something called sarcomeres. So let's talk about that a little bit more closely. For years and years and years, all we had was a microscopic image of what muscle tissue would look like. So you could cut it into a very fine slice, look at it under the microscope, and you would see these striations. But it's taken many, many years, lots of experimentation to kind of understand how those striations correspond to these contractile proteins. And of course, better technology with higher magnification microscopes. So essentially what it is the myosin and actin are organized in this way that I've drawn for you on previous slides, in which you have the Z discs on either end. You have actin filaments coming off of each Z disc. You can see the myosin in purple, so the thick filaments throughout the middle. You've got that M line in the middle. You've also got some elastin here connecting the Z disc to the myosin, so that's going to be in addition to where we talked about it before, being through the core of the actin and in the M line. And over the years, they've given names to these regions, and we'll talk about that in a minute. But essentially, from Z disc to Z disc is called a sarcomere, all right? And the sarcomere is the smallest functional unit of a muscle, so it's the smallest contractile unit. And the way this arrangement occurs, it explains the striations that we see when we look in the microscope. So what these striations are, and you can see here in the micrograph, we've got dark band, dark band, dark band, and then you've got a light band and a light band. And really, the lines within the light bands are the Z discs, and this is going to be a sarcomere then. So from the middle of one light band to the middle of the other light band, you have a sarcomere. And the dark region in the middle is the A band, and the I bands are known as light. This is what accounts for the striations that we see, is basically the arrangement of these myofilaments. So again, the A band is going to be the dark band, so it's where you've got the thick filaments overlapping, and there are some overlap with the thin filaments in this area too, so it's going to be darker. The H band is here in the middle. You can kind of barely make it out. It looks almost like a little light band in the middle of the A band. This is where it's only thick filaments. So you can see that down here in our schematic. That is going to be the H band. And then again, the A band is darker because it's got both myosin and thin filaments overlapping. The M line in the very middle is the dark transverse protein in the middle of the H band. The I bands, again, are the light bands, so that's where we have only actin, and it's going to include parts of two sarcomeres, because remember, one sarcomere is from Z disc to Z disc, but the light band is going to show when it's actin only, and so you've got the actin only of one sarcomere, the Z disc, and then the actin of the other sarcomere. And then the Z disc, of course, is that the protein that serves as the end of the sarcomere. And so it's got the anchorage for the thin and the thick filaments. Again, the sarcomere is the smallest functional unit of the muscle contraction. I like to show this a couple of times just because you're exposed to it more with different images. So again, we have these alternating dark and light bands, A and I, respectively. Remember, A, again, is going to be where you've got that overlap of the myosin and the actin. When you just have myosin, it's called the H band. An entire sarcomere is, again, from Z disc to Z disc. The muscle cells themselves are going to shorten because these sarcomeres shorten. So if you will, think for a minute, there are hundreds and thousands of these sarcomeres in each muscle cell, all right? And so when these sarcomeres shorten, because of the overlap increasing between myosin and actin, when they all shorten, then you are shortening the overall length of the muscle cell. in this shortening that we're talking about, the Z discs are pulled closer together and the thick and thin filaments are sliding past each other. Something that's really important to keep in mind, you've got these myosin heads that are binding onto the actin, and they're going to pull the actin further over them and closer to each other during the shortening. Neither one of these filaments themselves shorten. It is the amount of overlap that changes and the overall size of the sarcomere that's shortening. But the filaments themselves are not shortening. During the shortening, the dystrophin protein we talked about and the other linking proteins are going to pull on extracellular proteins. And so this is how that force of the shortening of the sarcomere is transferred to the other parts of the muscle.